Welcome. In today's class, we are going to learn about phase contrast microscope. It is always believed staining the organism or the specimen will enable us to visualize the organism much better. But at times when you want to visualize the living organisms, live tissues, or any such live host or a specimen, it is not possible to stain them. In such case, phase contrast microscope comes very handy where you can study the organisms alive, unstained. Here, we are going to use light, that is the source, which will enable us to study the organism effectively without staining it. As in, the live organisms can be studied, live condition of the specimen can be studied. Phase contrast microscope is an optical microscope which helps us to study the organism or the specimen without being stained in its transparent form itself with the help of light. Here, there is a phase shift that is created by the lens that are inbuilt in the phase contrast microscope, enable us to study the transparent specimen by changing the brightness in varied positions. Here, this is how the ray diagram of phase contrast microscope goes. Like here we have the light, that is the source, and there is a series of objective lens or the magnifying lens arranged. We have the annular diaphragm. Here this annular diaphragm has numerous annular grooves in that enables all the light to pass through it. The diaphragm has an ability to distinguish or to control the intensity of light that has to pass through the specimen. Condenser situated just above the diaphragm will enable to condense all the light that is coming from the diaphragm. Here, this condenser has no ability to control the intensity of the light, but just to narrow down all the light that is coming from the annular diaphragm towards the specimen so that no light is being wasted. After this is the specimen, when the light passes through the specimen, there is a production of two types of light. That is, the light wavelength or the pathway is being distinguished. The light gets differentiated as retarded light ray and unretarded light ray. Once it hits the specimen, there is a phase shift that is created, and that will enable us to quantify the phase shift. This phase shift is then converted to amplitude, and the difference in the retarded light ray and unretarded light ray will help us produce a final magnified image. Here, the objective lens will enable us to magnify the image, and then it is focused onto the faceplate. This faceplate has an ability to distinguish the or to quantify the retarded light ray and unretarded light ray, and then the final magnified image is produced. This is how the phase contrast microscope works. Look just like your compound microscope, but then the phase shift quantifier is present in this exclusively. But the phase shift is not generally visible through human eye, and that has to be quantified and then changed to amplitude, and then the difference has been observed as the image contrast. These are the parts of your phase contrast microscope, where there is an annular diaphragm. It is situated just below the condenser. It is made up of numerous circular annular grooves that allows the light to pass through it. Through the annular groove of your annular diaphragm, the light ray from the source enters into the condenser and then reaches the specimen. At the back focal plane of your objective lens, the final image has been produced. Annular diaphragm has numerous annular grooves that help us to control the intensity of light passing through. This is how the phase contrast microscope looks like, and these are the phase contrast objective lens, and then there is a condenser, and then there is a diaphragm here. So this working principle of the phase contrast microscope explains whatever we discussed in the diagram. partial coherent illumination, that is the light that is obtained as a source, is produced from the tungsten halogen lamp. That is from where we get the light. That is being collected and it is focused onto the specialized annulus diaphragm. That is your annular diaphragm. Specialized annulus is your diaphragm. And then it is proceeded to the substage of your condenser front focal plane. This is what we were discussing, where we have the halogen, tungsten halogen lamp that produces the light that hits the diaphragm, and then it is given to the condenser. Then wave front passing through the annulus illuminate the specimen, which means now the light hits the specimen. It either passes through undeviated or diffracted or retarded in pace by the structures and the pace gradient present in the specimen, which means now the light that has hit the specimen either gets converted as a retarded light ray or unretarded light ray. There are two types of light that will be produced after hitting the specimen, that is retarded light ray or unretarded light ray has been produced after hitting the specimen. Now, undeviated light and the diffracted light collected by the objective is segregated. Now, that quantification is done by your objective lens. Now, the segregation of your retarded and unretarded light ray occurs in your objective lens, which will help us to magnify the image. Once that is done, the face plate and the focused and the intermediate image plane to be formed at the final image after the quantification of the phase shift produces the final magnified image. So this diagram summarizes how the light influences us to produce a phase shift and how that phase shift has been quantified and how end of the day the final magnified image has been produced. Then this is the breakdown or a write-up on your annular diaphragm, and this is a write-up of your face plate. Face plate has two conjugates, or the conjugate area, that is a thick circular area or thin circular area. When it has a thick circular area, we call it a negative phase. When it has a thin circular area or a groove, we call it positive phase. This is a conjugate area where there is a thickening or thinning of its region or its groove, that is called as a conjugate. It can be a positive phase plate or negative phase plate that we can use in the phase contrast microscope. They are generally a transparent disc. These help the annular diaphragm and the face plate together produce as a final magnified image. The role of a face plate is to quantify the phase shift and produce as a final magnified image. So what are the applications and uses of phase contrast microscope? As told, the organism can be studied unstained, so it avoids the sample preparation. The organisms can be studied in its live condition. It helps us to study live cells, microbes, thin tissues, lithographic patterns, fibers, latex dispersion, and many other morphology study can be done. Its application is to study the organism in its just like the way it is, as we do not have a sample preparation. Here it is highly transparent objectives are visible so that the internal detailing is done. It has a very good resolution. Dynamic motilities can be studied. All the metabolism can be studied. Movements can be studied. So this is all about the phase contrast microscope. Thank you.